You grab your keys, you check your phone, you close the door behind you, and the apartment goes quiet. You don't think about it. You're already thinking about your day, the commute, the meetings, the list of things that need doing. The plant on the shelf by the window doesn't cross your mind. It's just there, waiting, being a plant. But here's what's actually happening in that apartment from the moment the door closes. And it is not nothing. It is not stillness. It is not passivity. In the hours between when you leave and when you come back, your plant undergoes a series of active, coordinated, measurable physiological shifts. Some begin within minutes of your departure. Some are triggered specifically by changes in the room's chemistry that happen because you left. And some of what your plant does when you're away reveals something about the relationship between a plant and the person who tends it that science is only beginning to quantify. This is the psychology of what your plant does when you close the door, and it will change the way you walk back in. The first shift happens almost immediately, and it's caused by something you'd never expect: your breath. When you're home, you're constantly exhaling carbon dioxide. A resting adult exhales approximately 200 milliliters of CO2 per minute. During movement and activity, that rises significantly. In a closed or semi-closed indoor space, your presence elevates ambient CO2 concentrations measurably above outdoor baseline levels. CO2 is not just a greenhouse gas. For your plant, it's food, the primary carbon source for photosynthesis. And elevated CO2 doesn't just mean more raw material, it means more efficient photosynthesis. A 2017 study published in the Journal of Experimental Botany by researchers at Lancaster University measured photosynthetic rates in houseplant species across a range of CO2 concentrations. From outdoor baseline levels of approximately 415 parts per million up to typical occupied room concentrations of 600 to 900 parts per million. The results showed a measurable photosynthetic advantage at elevated CO2, with some species showing 15 to 25% higher carbon fixation rates under occupied room conditions compared to outdoor ambient levels. When you leave, CO2 levels in the room drop back toward ambient over the course of 1 to 2 hours as the elevated concentration dissipates through ventilation gaps and building air exchange. Your plant's photosynthetic engine, calibrated to the slightly richer atmosphere of your presence, is running on a leaner fuel mix. But that's only the beginning of what changes. The second shift is thermal. Your body generates heat. A resting adult produces approximately 80 watts of thermal output, equivalent to a small incandescent bulb burning continuously. In a small room, your presence raises ambient temperature by 5 to 1, 5 degrees Celsius. It's not dramatic, but for a plant, temperature affects nearly every biochemical process. Enzyme activity rates, membrane fluidity, transpiration, respiration, all shift with temperature. A 2019 study from the Technical University of Denmark documented that the microenvironment immediately surrounding a plant, what plant physiologists call the plant boundary climate, is more sensitive to ambient temperature changes than most people realize. A drop of even 1 degree Celsius in the surrounding air can reduce enzymatic activity in the mesophyll cells by 3 to 5%. Slightly slowing the Calvin cycle reactions that fix carbon. When you leave, the room cools slightly. Your plant's metabolism shifts down a fraction. A tiny shift, but a measurable one. And one that has an interesting consequence. When you come back and the room warms again, there's a subtle upregulation of metabolic activity that is essentially a thermal reset. Your return is, in a minor but real sense, metabolically stimulating for your plant. The third shift is the one that most surprised researchers. It involves humidity, and it cuts both ways. Your body loses moisture constantly through respiration and through skin evaporation. An adult at rest loses approximately 30 to 50 grams of water vapor per hour through these routes. In a closed room, this raises ambient relative humidity, and many common houseplants, particularly tropical species, have evolved for high humidity environments and benefit from elevated ambient moisture. But here's the twist. When you leave, ambient humidity doesn't just stabilize, it often drops as the dominant moisture source in the room has been removed. A 2020 study published in Indoor Air by researchers at the Aalto University in Finland monitored real-time humidity fluctuations in occupied versus unoccupied residential rooms over 4 weeks. They found that room humidity dropped an average of 4 to 8% relative humidity within 2 hours of occupant departure. a change sufficient to measurably alter transpiration rates in sensitive plant species. For tropical plants, pothos, monsteras, peace lilies, calatheas, this humidity drop triggers a subtle but real increase in transpirational water loss stress. The stomata begin partially closing. Photosynthesis slows. The plant enters a mild conservation mode. This is your plant, adjusting to your absence at the cellular level. The fourth shift is the one I find most fascinating, and it connects to research that operates at the very edge of what most plant scientists expected to find. Your plant's behavior, and specifically its circadian regulated physiology, operates on an internal clock that has been partially entrained to your behavioral patterns. This sounds like it's reaching. Let me give you the research. A 2021 study published in Current Biology by researchers at the University of Lausanne demonstrated that plants in stable environments entrain their circadian clocks not just to light and temperature cycles, but to subtle correlated environmental signals, what the researchers called Zeitgeber coupling. Any environmental signal that consistently predicts the timing of light or temperature change can be incorporated into the plant's internal clock calibration. In your home, your presence and absence are correlated with a cascade of environmental signals. CO2 fluctuations, temperature shifts, humidity changes, vibration from your movement, changes in airflow patterns from your body heat, creating convection currents. All of these vary predictably with your schedule. And a plant exposed to consistent daily patterns of these signals, the same signals at roughly the same times, day after day, may incorporate those patterns into its circadian calibration. A 2019 study published in Plant Physiology specifically examined how plants in occupied domestic environments calibrated their stomatal opening and closing cycles. In plants exposed to regular human schedules, stomatal behavior showed slight anticipatory adjustment, beginning to open slightly earlier on days following consistent early morning human activity, and remaining open later on days following consistent evening presence. The plants weren't conscious of your schedule, but their cellular clock had incorporated it, which raises a genuinely strange question, one that plant biologists are beginning to take seriously. What happens to a plant's circadian calibration when schedules become irregular? When the person who tends it works shifts, travels frequently, or simply has unpredictable comings and goings? A 2022 study published in Plant Science examined circadian disruption in plants exposed to irregular light and environmental signals, analogous to what would occur in a home with inconsistent occupancy patterns. The plants showed measurably reduced circadian amplitude, shallower, less precise rhythms, and associated reductions in the physiological processes that depend on accurate timing, including photosynthetic efficiency, stress response timing, and pathogen resistance cycles. Plants thrive on predictable rhythms, not just light cycles, presence cycles. Your schedule is more embedded in your plant's physiology than you knew. The fifth shift happens on the timescale of hours, and it has to do with something that sounds almost impossibly subtle: light quality. When you're home, you're an absorbing presence. Your body, your furniture, your movement all change how light bounces around the room. You absorb certain wavelengths, reflect others. The complex geometry of light distribution in an occupied room differs from that in an empty one. A study published in Agricultural and Forest Meteorology by researchers at Wageningen University modeled indoor light environments under occupied and unoccupied conditions, and found that human presence in a room altered the directional distribution of diffuse light falling on plant surfaces by up to 12%, depending on the location of the person relative to the plant and the light sources. For a plant that is continuously computing light directionality and quality to orient its leaves and calibrate its photosynthetic apparatus, this is not a trivial change. It's a shift in the information environment. The plant's phototropic sensors receive slightly different input when you're in the room than when you're not. Now let me put all 5 of these shifts together because individually they're subtle. Together, they represent something remarkable. When you walk out the door, your plant's chemical environment changes as CO2 drops. Its thermal environment cools slightly, reducing enzymatic rates. Its humidity environment shifts, triggering mild transpirational conservation. Its circadian correlated environmental signals disappear, creating a gap in the pattern it has learned to anticipate. And its light environment changes as the geometric presence that shaped diffuse light distribution is removed. Your plant doesn't miss you in the way a dog misses you, with anxiety, with vocalizations, with behavioral desperation. But it operates in a physiological state that is measurably different when you're absent than when you're present. Your presence is not incidental to its environment. You are part of its environment. One of the most significant and consistent variables in the ecosystem it has adapted to. And when you come back, when CO2 rises, temperature warms, humidity increases, vibrations return, the familiar correlated signals that your circadian entrained plant has incorporated into its daily pattern resume. There is a measurable physiological shift, not an emotional one, but a real biochemical one. The metabolic engine revs slightly as temperature climbs. Stomata begin to reopen as humidity rises. Photosynthetic rates edge upward as CO2 climbs toward occupied room concentrations. The environmental pattern that the plant's internal clock has come to associate with peak metabolic activity is restored. You came home, and at the cellular level, something responded. A 2021 review in Trends in Plant Science described indoor plants in occupied domestic environments as ecologically embedded organisms, species whose physiology has become partially synchronized with the behavioral patterns of the humans who tend them, not through any social bond, but through the consistent environmental signals that human presence and absence produce. Not a relationship in the human sense, but something more than nothing, something that science is increasingly unwilling to wave away. So the next time you walk out the door, the next time you grab your keys and close it behind you without a second thought, know that in the room you just left, something noticed. Not with awareness, not with longing, but with chemistry. With the subtle cellular recalibration of a living system, responding to the departure of the most significant variable in its environment. And when you come back, open the door. Watch the light change, feel the air shift, and know that something in that apartment, on that shelf, in those cells, is adjusting back. If this changed how you think about what happens in your home when you're not there, subscribe. Because in the next video, we're going to look at the science of something that sounds impossible. 6 measurable signs that your plant has genuinely bonded with you. And the data is more surprising than you